Today to discuss cellular responses to stress and toxic in cells. So this is part 2 of the lecture that we started during the previous day. So we are going to focus on the following issues. We'll talk about subcellular responses to injury, intracellular accumulations, pathologic calcification. But before we'll start discussing all these important issues, we have to talk about basic subcellular constituents or components existing in the cell. On the picture, you see the number of various organelles within a virtual cell. The figure shows relationships, but is not intended to be accurate to scale. The stresses and the injury affect the components of the cell and organelles inside it, and functional status of cells and tissues. The cell is a universe, where the nucleus is the sun, while all other organelles could be considered as planets. Every cell contains nucleus, the organelle that contains the cell's DNA and is the control center of the cell. You also can see on this picture mitochondria, the organelle that breaks down food molecules to make ATP, endoplasmic reticulum, the organelle that makes lipids, breaks down drugs and other substances and packages protein for Golgi complex. Obviously, there is a Golgi complex that processes and transports proteins and other materials out of cell. There are ribosomes, organelles in which amino acids are hooked together to make proteins. There are lysosomes that digest food particles, wastes, cell parts, and foreign invaders. The cell nucleus, shown by the red star, is the most important and defining feature of the cell. Most of the genetic material, also known as DNA, is contained in the nucleus, while a small amount of it is found in mitochondria. The nucleus has a very important role to play. As it contains genetic material, it coordinates cell activities like protein synthesis and cell division. The nucleus is made up of several components: nuclear envelope, nuclear lamina, nucleolus, chromosomes, nucleoplasm. These are only several components that play a very important role. All of these components work together in order for the nucleus to accomplish all its functions. These functions include control of the genetical information of the cell and thus the hereditary characteristics of the organism. The functions also imply control of the protein and enzyme synthesis, control of cell division and cell growth, storage of DNA, RNA, and ribosomes, regulation of the transcription of mRNA to protein, productions of ribosomes. The stresses and the injury can affect nucleus, and functional status of cells can change. There are several examples of nucleus diseases. Laminopathies. Mutations in the genes that code for lamins in the nucleus can lead to a number of rare genetic disorders, normally due to a change in the abundance of the lamins in the nucleus. Collectively, these diseases are known as laminopathies. These diseases include autosomal dominant Emery-Dreifuss muscular dystrophy, Danon type familial partial lipodystrophy, as well as developmental and aging disorders. Nucleus abnormalities. Additionally, involve certain blood disorders that can lead to abnormalities in the nucleus. That means that analysis of the shape and structure of nuclei in blood cells can lead to diagnosis. For example, Wilson disease leads to an increase in glycogen in the nuclei, whilst acute myeloid leukemia causes nuclei to become cup-shaped. Moreover, abnormalities in the nucleoli can lead to some forms of rare hereditary diseases, as well as degenerative diseases, such as Huntington's and Alzheimer's. Several diseases can also result from changes in the nuclear envelope. These include cardiomyopathy and muscular dystrophy. Some examples of these diseases that were mentioned you can see on this slide. Another important organelle in the cell is a mitochondrion. Mitochondria, shown by the yellow star, are well known as the powerhouse of the cell because mitochondrion plays a role in the production of ATP. Pyruvate molecules produced by glycolysis are actively transported across the inner mitochondrial membrane. Protons can re-enter the mitochondrial matrix without contributing to ATP synthesis. Mitochondria can transiently store calcium, a contributing process for the cell's homeostasis of calcium. The relationship between cellular proliferation and mitochondria also is extremely important, and mitochondria plays a central role in many other metabolic tasks, signaling for mitochondrial reactive oxygen species, regulation of the membrane potential, apoptosis programmed cell death, regulation of cellular metabolism, certain heme synthesis reactions, and others. We know several mitochondrial diseases, including mitochondrial myopathy, diabetes mellitus and deafness, Leber's hereditary optic neuropathy, Lyme syndrome, subacute sclerosing encephalopathy, neuropathy, ataxia, retinitis pigmentosa, and ptosis, myoneurogenic gastrointestinal encephalopathy. As mentioned earlier, mitochondrial dysfunction plays an important role in acute cell injury and death. In some non-lethal pathologic conditions, however, there may be alterations in the number, size, shape, and presumably function of mitochondria. For example, on the right, you can see cellular hypertrophy, and there is an increase in the number of mitochondria in cells. Conversely, mitochondria decrease in number during cellular atrophy, probably due to autophagy. Mitochondria may assume extremely large and abnormal shapes, and this process is known as megamitochondria, and it is signified by red stars, as seen in hepatocytes in various nutritional deficiencies and alcoholic liver disease. Endoplasmic reticulum is another component existing in the cell. It serves multiple functions, being important particularly in the synthesis, folding, modification, and transport of proteins. The endoplasmic reticulum usually constitutes more than half of the membranous content of the cell. Differences in certain physical and functional characteristics distinguish the two types of endoplasmic reticulum, known as rough endoplasmic reticulum and smooth endoplasmic reticulum. On the right, you can see rough endoplasmic reticulum. On the left you can see rough endoplasmic reticulum, and it is shown by the white stem, and smooth endoplasmic reticulum on the right is shown by the yellow stem. Rough endoplasmic reticulum is named for its rough appearance, which is due to the ribosomes attached to its outer cytoplasmic surface. The ribosomes on rough endoplasmic reticulum specialize in the synthesis of proteins that possess a signal sequence that directs them specifically to the endoplasmic reticulum for processing. Smooth endoplasmic reticulum functions differ in a different way. The smooth endoplasmic reticulum is involved in the synthesis of lipids, including cholesterol and phospholipids, which are used in the production of new cellular membrane. Endoplasmic reticulum dysfunction might have an important part to play in a range of neurological disorders, including cerebral ischemia, sleep apnea, Alzheimer's disease, multiple sclerosis, amyotrophic lateral sclerosis, the prion diseases, and familial encephalopathy with neuroserpine inclusion bodies. Lysosomes, shown by the yellow star on the left, are membrane-bound organelles which contain, among other components, hydrolytic enzymes that operate in an acidic environment. Lysosomes are capable of digesting all types of macromolecules and participate in the breakdown of both extracellular and intracellular components that are targeted to them through the processes of endocytosis or autophagy. Lysosomal diseases, also known as lysosomal storage diseases, are a heterogeneous group of diseases caused by defects in lysosomal function. Most lysosomal diseases result from a deficiency in lysosomal hydrolases, including alpha-galactosidase in Fabry disease. Alternatively, lysosomal diseases may be caused by deficiencies in lysosomal membrane-associated transporters, including cystosine in cystinosis, or other non-enzymatic proteins, including CLN3 in Batten disease. In the lysosomal diseases associated with enzyme deficiencies, diagnosis is usually created by the direct measurement of the activity of the enzyme associated with the disease. We are continuing our discussion of the functional capacity of lysosomes. Autophagy on the left refers to lysosomal digestion of the cell's own components and is contrasted with heterophagy on the right, in which a cell, usually a macrophage, ingests substances from the outside for intracellular destruction. Autophagy is thought to be a survival mechanism in times of nutrient deprivation, such that the stuffed cell lives by eating its own components. In this process, intracellular organelles and portions of cytosol form an autophagic vacuole, shown by the yellow star on the left part of the scheme. The vacuole fuses with lysosomes to form an autophagy lysosome, shown by the red star, and the cellular components are digested by lysosomal enzymes. Lysosomes with undigested debris may persist within cells as residual bodies shown by the black star on this figure. The Golgi apparatus is another component of the cell. It is a membrane-bound organelle. It is responsible for packaging proteins into vesicles prior to secretion, and therefore Golgi apparatus plays a key role in the secretory pathway. The Golgi apparatus modifies proteins and lipids that it receives from the endoplasmic reticulum. These biochemicals leave the Golgi by exocytosis before being delivered to different intracellular or extracellular targets. Dysfunction of the Golgi apparatus leads to the development of Wilson's disease. Wilson's disease is an autosomal recessive disorder characterized by an abnormal accumulation of copper in the body. The liver is particularly susceptible to this accumulation. Patients experience symptoms and signs of liver failure, such as itching, abdominal swelling, fatigue, jaundice, ascites, and Kaiser-Fleischer's rinse that implies depositions of copper in the cornea. They may also present with neuropsychiatric symptoms such as confusion, psychosis, personality changes, and tremor. Copper may also accumulate in joints, causing joint pain and swelling. Obviously, there are other important organelles existing in the cell, and they include cell membrane that controls what enters and what leaves the cell, cytoplasm that represents a jelly-like fluid that fills the cell between the cell membrane and nuclear membrane that holds all organelles in place and is responsible for the movement of organelles around the cell, and centrioles that play a role in cell division. Nucleolus also is important because it is responsible for making mRNA and ribosomes. The cytoskeleton consists of actin and myosin filaments, microtubules, and various classes of intermediate filaments. Several other non-filamentous forms of contractile proteins also contribute to the cellular scaffolding. The cytoskeleton is important for many cellular functions: intracellular transport of organelles and molecules, maintenance of basic cell architecture, including cell polarity distinguishing up and down, transmission of cell-to-cell and cell-to-extracellular matrix signals to the nucleus, maintenance of mechanical strength for tissue integrity, cell mobility, and phagocytosis. Therefore, cytoskeletal abnormalities might lead to abnormal appearance and function of cells, including hypertrophic cardiomyopathy, aberrant movements of intracellular organelles, defective cell locomotion, intracellular accumulations of fibrillar material as in alcoholic liver diseases, perturbations in the organizations of microtubules as a cause for sterility by inhibiting sperm motility, and defective motility of cilia in the respiratory epithelium, like in Kartagener syndrome or the immotile cilia syndrome, and others. I would like you to suggest a case study of Kartagener syndrome. Kartagener syndrome is an autosomal recessive genetic disorder. The syndrome includes the clinical triad of chronic sinusitis, bronchiectasis, and situs inversus. Ciliary dyskinesia is the cause of the Kartagener syndrome. Immotile cilia syndrome is also a synonym of Kartagener syndrome. And normal ciliary function is critical for respiratory host defense and motility of sperm. In Kartagener syndrome, the gene mutation at DNA L1 and DNA H5 leads to impaired ciliary motility, which predisposes to recurrent sinopulmonary infections and infertility. So we are looking at the case of the 24-year-old man who has Kartagener syndrome. He was presented with the complaints of recurrent episodes of nasal congestion with itching and paranasal discomfort and productive cough for almost more than a decade. He noticed frequent exacerbation of cough with copious purulent sputum in the last 3 years. He was treated for pulmonary tuberculosis 7 years back as smear-negative pulmonary tuberculosis, but there was no significant clinical improvement after completion of 6 months anti-tuberculosis therapy. A chest-computed tomography scan showed bronchoectatic changes prominent on both lower lung fields. Ultrasound examination of his abdomen showed liver and inferior vena cava on left side and spleen on right side, suggestive of situs inversus. A diagnosis of Kartagener syndrome was made on the basis of clinical presentation, imaging features, and biopsy. Electron microscopy, lack or dysfunction of Denian arms showed lack or dysfunction of Denian arms, radial spokes, and microtubules of cilia. They were recognized structural and functional abnormalities of ciliary ultrastructures. Abnormalities of ciliary ultrastructures encoded by the mutated genes DNA I1 and DNA H5. These faulty genes cause the cilia to be rod-shaped, not round, and this is shown by these red stars on the picture. Or the cilia can also move in a wrong way, making ciliary motility defective. That is why all stresses and noxious influences exert their effects first at the molecular or biochemical level. And that is why you need to understand that cellular functions may be lost long before cell death occurs, and the morphologic changes of cell injury or death can lag far behind the mentioned causes. On this picture, you see the relationship between cellular function, shown by the blue star, cell death, shown by the yellow star, and the morphologic changes, shown by the white stars, of cell injury. You understand that cells may rapidly become non-functional after the onset of injury, although they are still viable with potentially reversible damage. A longer duration of injury may eventually lead to irreversible injury and cell death. Remember that cell death typically precedes ultrastructural light microscopic and grossly visible morphologic changes. For example, myocardial cells become non-contractile after 1 to 2 minutes of ischemia, although they do not die until 20 to 30 minutes of ischemia have elapsed. These myocytes do not appear dead during electron microscopy for 2 to 3 hours, and by light microscopy examination, they do not appear dead for 6 to 12 hours. Look on your right, and on the top picture you see the white star, and you can see early phase of acute myocardial infarction, and cells shown during the light microscopy, they look like normal cells. It is absolutely impossible to find cells with morphological signs of cell death. But on the other picture, pay attention to the black star. You find all morphological signs of cell death: coagulative necrosis, karyopyknosis, and karyorrhexis, the issues that we discussed during the previous class. Cell death resulting from lack of oxygen and necrotic cells show increased eosinophilia, pink staining from the eosin dye, the eosin in hematoxylin and eosin, and this is attributable in part to increased binding of eosin to denaturated cytoplasmic proteins, and in part to loss of the basophilia that is normally imparted by the ribonucleic acid RNA in the cytoplasm. Basophilia is the blue staining from a hematoxylin dye, the H in hematoxylin and eosin. We are going to stop here, and I will upload the second part of this lecture in a bit.